start learning about non-Mendelian inheritance. So there are a bunch of different inheritance patterns. And so previously, we've been focusing on Mendelian inheritance, right, which is simple dominance. There's one gene, two alleles, right, capital B or lowercase b, and two phenotypes, black or brown, right, as just an example of what we've been focusing on. But that is not always the case. So examples of those simple dominants that we've previously been working on is a widow's peak, right, which is like when your hairline goes like that, a cleft chin, right, like a butt chin, tongue, that's a horrifying picture, and then your earlobes, right, so this is a free earlobe and an attached earlobe, right, so it's one gene, you can have A or B, etc. And then, right, and here we have our cattle, our hornless cattle, or our non-hornless cattle, right, and it's still just one gene. But not everything is like that. Not everything fits that mold. So the first sort of abnormal thing we have going on is something called incomplete dominance, right? So that means that there's still just one gene and two alleles, right? But that the dominant allele is not completely dominant, right? So if you think, this is sort of like a halfway in between, right? So your recessive is still the same, and your dominant is still the same, but when you get to your heterozygous, things get different. So instead of two phenotypes, you now have three possible phenotypes. So let's take a look at what that looks like. So here's an example of that. So what's going on here, right, so your three phenotypes are red, pink, and white. So in this example, your red is fine, it's the same, right, and your white is fine. Those homozygous phenotypes are totally the same. It's just when you get to heterozygous that things get different. And that's because the dominant allele, right, that capital A is not completely dominant. So you're going to get an in-between trait. And if you think about this in terms of proteins, this red color is produced by a protein. And the white color means that none of that protein is being produced, right? So because there's no capital A, so there's no way you can produce that red protein. But when we go to here, this incomplete dominance is showing that about 50% of the red protein is being produced, and the other half of it is just white, right? So you get mixed results. Because you have to remember that these alleles, these A's, represent actual DNA sequences, which are translated to proteins, right? So in this case, you're getting two red proteins made, 100% red. In this, you're getting no red proteins made, 100% white. And this, you're getting 50% red proteins made, right? So it doesn't completely override. You get a mixed. And that's what's called incomplete dominance, right? It's different than what we've been learning about. To go alongside incomplete dominance is something called codominance, right? And codominance is where you have one gene, and you have two alleles, and both alleles are dominant, which means they're both represented with capital letters, right? And as a result of both of those alleles being dominant, you can see both traits, right? So we'll look at an example of that. So we're back to the flowers, and what you have seen here is an example of this codominance. So you have two dominant traits. You have a red color and you have a white color. And so the genotype of this individual, so a red individual, a completely red flower would be RR. A completely white flower would be WW. So you're getting into the two different letters. Again, this is different from what we've learned. And notice that all of these alleles are dominant, so they're all capital letters. So when you have this mixture, right, the genotype is actually going to be RW. because you have a dominant and a dominant allele. And so if you think about it in terms of protein, this capital R produces a red protein, and this capital W produces a white protein. So when both are produced at the same time, you get a spotted red and white individual. I hope that makes sense. Okay. Right, so here's another example. It's not necessarily just flowers. Sometimes it can be spots on a cow, right? So you have the red protein that's being produced and the white protein that's being produced, both dominant, so they're both at the same time. They're both produced and shown at the same time, right? So this would again be that RW phenotype, right? Heterozygous, co-dominant, two forms of dominance. Okay, so this is a comparison of incomplete dominance or co-dominance. Sort of the quick and easy way to tell the difference is that incomplete dominance is a mixture, right? It's pink, a mixture of those two, because you're gonna see this phenotype, right? Because this dominant allele is not completely dominant. It's like you're only seeing 50%, right? Whereas codominance, you're seeing spots, right? Or a pattern, and the genotype is two dominant traits, and you're seeing both of those dominant traits, both white protein and red protein is being produced in various patterns. Okay. Another example of this is specific diseases. It's not always colors. It can be all sorts of things in the body, right? Most traits in your body are not simple dominance, right? Most things in your body are not free earlobes, attached earlobes. Like, that's too simple for a lot of things. So one example of codominance, right, is this idea of sickle cell diseases, right? So if it's AA, they're all round cells. And if it's SS, they're all sickled cells. This is a sickled cell, right? It just means it's kind of like twisted. And sometimes people have a codominant genotype where there's half round cells and half sickled cells, right? And sickled cell can be a really dangerous disease, but it also can give you side effects. If you have all round cells, you can get malaria. If you have all sickled cells, you do not get malaria, right? So there's a benefit to this. There's a big benefit in the sense that if we look at your cells, right? If you have all round cells, you're not getting the disease, but you can get malaria, a separate disease. Whereas if you have all sickle cells, right, you're going to get sickle cell disease, which is bad. We don't want that. But you can't get malaria, this separate disease. But if you're in the middle, it's the best of both worlds. If you have that codominance, you can get you're not going to get the full disease, right? You're only going to get a little bit sick. And you're going to prevent yourself from getting this malaria infection, this other disease. So this is sort of the best of both worlds because you don't get malaria and you don't have sickle cell disease in full force. So it's sort of another example of seeing codominance in real life, right? It's not always just colors. There's lots of other things. Okay. Okay, so now there's some other examples of things. So we've covered incomplete dominance and codominance, and then some things in your body have multiple alleles. And let's remind ourselves what an allele is, right? So an allele is that DNA sequence, right, from mom or dad. So maybe dad gave you capital A. Ooh, let me freeze that. Maybe dad gave you capital A and mom gave you lowercase a. Those are two different DNA sequences. And in your body, they come together, right, and create a phenotype of, I don't know, what could your phenotype be? Brown eyes, let's say. Right? So these alleles are DNA sequences, and most things, or a number of things in your body are done with just 2 alleles, right? One from mom, one from dad. But there's something unusual that can happen. Sometimes there are different versions of these alleles. So again, you're only ever going to have 2, right? Because there's no third parent, but you might have a capital 1 and lowercase 2, right? So or you could have this combination. So there's some more subtle differences in your alleles, and that can determine lots of different things. For example, one of the things that's a really common example of multiple alleles is blood type, right? So if we look over here at this example, if you have these 2 alleles for blood, you're going to have type O blood. If you have this combination of alleles or this combination of alleles, you'll have type A. Notice how this is different than this, right? And then down here we have a third allele, IB, right? So this is 1, 2, 3 different types, and that creates 4 different blood types, right? So you can see that there's 3 alleles. There's only ever going to be 2 at a time because, again, you do not have a third parent, right? Maybe in your life you have someone who's like a third parent, but there were only ever 2 people that biologically created you, right? So there's only 2 parents. So you can have all these different combinations, but sometimes there's more than 2 options, right? There's more than just capital B, lowercase b, right? We have more than 2 options. So blood type is a really common example for that. Okay. The alternative to having more than 1 allele is that some traits have more than 1 gene, right? If you think about a good example is height, right? There's not one thing that determines your height. What determines your height is the length of your bones, right? And that's run by many, many different genes. It's not like you just have the tall gene, right? There's thousands of genes that affect height. There's thousands of genes that affect eyesight, right? That's why you can be a wildly different height than your parents, because there's just different genes that can affect that. And environmental factors, right? Don't drink a lot of caffeine when you're young. You won't grow. So polygenic traits means multiple genes. Poly meaning many and genic meaning genes. So many genes, right? So we'll look at an example of this. Another example is skin color, right? So there are many different genes involved in determining your skin color. Right, this example is these 3 genes, A, B, and C, are involved in determining skin color. So you can have really light skin, right? Or you can have really dark skin, and it's a combination of the phenotypes and genotypes of all 3 of these genes, right? And there are more than 3 genes. This is just a simplified example. So a lot of things about your body are affected by more than 1 gene. Most traits in your body are polygenic, right? How big your liver is polygenic. A lot of your brain development is polygenic, right? It can't be as simple as 1 trait. Hair curliness is polygenic, right? And then finally, we have something called epistasis, right? So epistasis is this idea that if you have 1 specific genotype, that can block out everything else, right? So a great example of this is Labrador dogs, right? So you can be capital B, black, or lowercase b, chocolate, but you see all these yellow labs. How is that possible? Well, there are 2 genes, right? So you can have this B gene and the E gene involved, and if the E is not recessive, then everything's fine, right? So then if the E is not recessive, then we just care about the B gene. And that says this is black, black, black. So all of these genotypes are going to be black phenotype. Brown phenotypes, again, as long as the E gene is not recessive, homozygous recessive, we don't care about it, and we just focus on the B, right? And this says this is chocolate. But if the E gene is homozygous recessive, right? If you have two lowercase e's, that overrides everything else. It blocks everything else. And all of a sudden, all of these, no matter what the B is, become yellow for their phenotype. And so in a sense, this yellow is, we call this epistatic, right? So because it blocks everything else, right? It overrides all the other things when it is homozygous recessive. And there are many other examples of this as well. Certain genes will override all the other signals in your body, right? So here's another example of this, right? So no matter what, brown is dominant, brown eye color is dominant over all the other eye colors, right? So brown will be dominant whether you have blue eyes or green eyes, right? It's epistatic. It is always going to be dominant. Okay, and that is it for non-Mendelian modes of inheritance. Next lesson, we will begin learning about sex-linked, and then we'll finish up with dihybrid, and we're going to learn a little bit more about some other genetic components, like some genetic diseases. But for now, just make sure you answer the questions on this worksheet attached to this lesson.